process. So essentially in cancer oncology area, genomics is used in a big way. Now, next we have pharmacogenomics. Who has heard of pharmacogenomics? It, okay, has. Okay, it's actually an emerging field. And the reason is, what has made it possible is that we now have quite a bit of genomic information, right? And it's using genomic information or the genetics of humans to answer why there's so much variation in how human populations respond to therapeutics. That's very important, right? We don't all respond to therapeutics, drugs, treatments the same way. And that's pharmacogenomics. Then another very useful application of genomics is prenatal testing. And that is being used in a big already. So essentially, to make sure if you're pregnant mothers, if there are certain genetic diseases, etc. So they would screen for those. So gene therapy is another one. Disease risk prediction, and this we've seen for a long time, especially, for instance, with breast cancer genes, etc. So population health and epidemiology, and the last one, biomarker discovery. So again, there are far more uses of genomics outside of infectious disease, and this is sort of some of them. Now, we were talking more about DNA sequencing there, right? But there's even other types of genomics that we haven't even touched upon. So when we talk about genomics, we were essentially talking about genomics here. I don't know if you can see the cursor. But actually, genomics has, you can do what's known as epigenetics, right? Because DNA also has certain modifications that also falls under genomics. You can also look at RNA expression, gene expression, that also falls under genomics, so that's transcriptomics. You can even sequence out the microbiome. So that's not the pathogen, right? Here we're not talking about pathogen genomics. We're actually talking about your microbiome that either lives in your gut, your skin, your nasal mucosa, and 16S sequencing is one of the ways. And again, that's another type of genomics. And you can also integrate these different kinds of genomics, and which is known as multi-omics. So it gets more and more complex. But in fact, it gives us new and newer tools to work with, right? For disease prediction and all the things that I showed in the earlier slide. But how can we apply this in low resource settings? So again, the use case that I have is for pathogen genomics. So I'm just going to go through a study that we did when we started setting up Asia PGI, and essentially some of the barriers that we saw for setting up an advanced technology in low middle income countries, they hold true for a lot of the other advanced technologies in genomics. So this was very much, so again, this is just a use case. This was an end-to-end national level assessment of the, when I say current status, priorities, and bottlenecks in pathogen genomics, it was back in 2023. So how we started was we essentially set up a survey, and we looked at what was existing in terms of questionnaires that people had used to look at NGS capacity in countries. And we tweaked it, so there are quite a few existing already. A lot of it were very lab-based. They weren't looking at national capacity. And essentially we took them, we looked at each of those surveys, and then we used them as reference documents to develop our own questionnaire. And the framework that we used was, we realized there were 3 areas to look at. There was one, the enabling environment, right? Which involved who are the partners who are needed in setting up this advanced technology, the financing options, the policy guidelines, the national status. Then the capacity, and the capacity includes laboratory infrastructure, it includes the bioinformatics infrastructure, it also includes our supply chain. And finally, quality assurance and data sharing. And essentially we went through this process this was actually several years of, you know, generating this tool, validating, you know, having experts look at it, see is this the right kind of tool or questionnaire, then taking it to country partners. They looked at it and they looked at whether the questions were relevant for them. And then finally, we refined the tool and we did the landscape assessment. So the questionnaire had over 100 questions and 19 pages long, and with all the sub-questions, it was 250 plus. So, but I'm not showing you all that data. In fact, I don't think we've analyzed all that data, to be honest. So these are the initial partners we had starting off with the Asia Pathogen Genomics Initiative, and these were the countries and partners that we worked with. And the reason we worked with these partners for this specific question is because we looked at who were the partners uploading data, pathogen genomics data, to GISAID. And we realized that these partners together, except for Myanmar, I think Myanmar, it's a bit of a different situation. The partners that we worked with essentially accounted for more than 50% of the SARS-CoV-2 data that was uploaded to GISAID. So essentially, the main sequencing, pathogen sequencing partners at the time. And majority of what was sequenced at the time was SARS-CoV-2. And so what we realized was in our region, majority of the capacity for sequencing for pathogens at the time was between the public sector and the academic sector. So those were the two main groups. And in some countries, they worked really well together. Some countries not so much, but most countries, they did work well together, especially during COVID. Now, the areas, they did often get external support, and that tended to be in areas of data analysis, bioinformatics training, lab training. There was a lot of donation of equipment and reagents. This is during COVID, right? And there was a lot of financial and in-kind contributions. Then we asked, I go through this quickly because this is another challenges part. I think the part that we want to get into are the challenges. We did ask questions around what are the priority pathogens for sequencing in our region. It's interesting because this study was funded by Gates Foundation, and you know, Gates has their priority pathogens, and they assume that those were a lot of the priority pathogens that would come up. But actually in Asia, a lot of our priority pathogens are a little bit different from in Africa, for instance. So as you can see, obviously at the time, coronaviruses came as number 1 because that was during COVID. Then TB came as number 2. So for those colleagues who are working on TB is a big problem in our region. Influenza, no doubt, arboviruses, so that came as a big one, and bacterial AMR, so AMR. And between South Asia and Southeast Asian countries, there are only very minor differences, all pretty much similar ones that pop up. Then we asked the question, what purpose, you know, were these sequencing, did it happen in the human sector or the environment sector? We did ask whether it happened in the animal sector, but that wasn't relevant, so we don't show that data because we didn't have animal sector partners, so it wasn't really relevant. So, but as you can see, majority of the sequence, we also then asked, in which setting is this sequenced? Are you sequencing when there's an outbreak? Are you sequencing for routine surveillance? Or are you sequencing when there's research? And as you can see here, majority of it is in clinical surveillance, and majority of it tends to be for research purposes. Now, this is not surprising, right? Because genomics was a technology, or is still in many parts a technology that resided in academic settings, in research setting. It's just that during COVID for pathogens, it's moved very quickly into the public health sector. And now, as you can see, it's slowly moving into the clinical sector. In human genomics, it has actually already moved into the clinical sector, right? Into the healthcare sector, especially in oncology, prenatal screening, etc. Then we did ask the question of how many countries are doing sequencing for novel pathogens, especially metagenomics, which some of you heard about this morning. Um, Almost every country at the time was, except Sri Lanka and Brunei, but I believe right now they're also by now doing metagenomics in humans. Okay, and we then wanted to look at the capacity of sequencing in terms of how many sequences are they releasing per million population. And as you can see here, there's a wide variety, right? There's a huge range. With some countries, I think the top one, oh, there's no Singapore here, but okay, because Singapore is off the scale, I think. We're sequencing way more than our population. But as you can see here, there's a huge variety in the region in terms of how many SARS-CoV-2 sequences were sequenced in relation to their population, which is, again, an issue. Variety, range, diversity in the capacity of sequencing. Now, another important point we found was which we call efficiency, that is sequencing output versus actual sequencing capacity, and that we find that it ranges across the board. And obviously, you can find Indonesia here somewhere, I believe. Indonesia does have a lot of sequencing machines, so there is a lot of sequencing capacity in terms of machinery, but I think a lot of it is not used to full capacity for other barriers, right? There are other barriers which we'll go through. Then timeliness. So if we're talking about using genomics to make an impact on health, especially, let's say, genomics for outbreak purposes or genomics for clinical purposes, we need to make sure the data is turned around quickly, right? It cannot be turned around, let's say, in a month. But actually what we found was, for our region, at least the 13 countries that we did, to go from sample taking in a patient to a sequence reported, it went from 8 days to 113 days, the range. And on average, it was about 29 days. That's a lot. That's 1 month, right, on average. And you can see how that breaks down. So actually what we found was that the longest time was from sample collection to getting a sequence out. From sequencing, doing the bioinformatic analysis to reporting, it didn't take very long. But somehow that initial step takes a long time. And for those of us who's in the lab, we know why that takes a long time, right? Samples, one, need to come from very far in most of these countries because they're going to reference sites. And other one, we batch samples, right? We're not going to sequence one sample or two samples. We're going to batch about 96 at the time for SARS-CoV-2. So that's a challenge, right? If you start sequencing in your country and you don't have enough samples coming in a timely manner, you can't get the data turned around quickly to make an impact. So this is GSAID data, actually. GSAID used to show how much data sharing was happening across our setting. Okay, Singapore shared a lot of data. Okay, that's fine. Even for data sharing, there's quite a variety or diversity in terms of which countries are sharing more data than others, and it doesn't quite always reflect the population size. It doesn't correlate with population size necessarily. And essentially, why is this important? Why is data sharing important? Anyone? Why do we want people to share data, especially genomics data? Maybe the group that was in the sequencing team this morning? Correct, correct. You want to hear that? He said to establish the database. So, so the thing with genomics is one sequence is not sufficient, right? To actually maximize the use of it, you need a whole database of information. And against that database is what people usually check their newly sequenced data against. And if that database, so that database is created with data, right? So if no one's sharing data into that database, then actually everyone loses because no one can do a proper analysis. No one can draw their phylogenetic trees, do their risk factor analyses, etc. So then we ask the question, what type of data are the countries that are sharing data sharing? Majority of the countries are sharing in the form of de-identified metadata and genome sequencing data. And even a few countries are sharing, about 39% are sharing raw FASTQ files. Now, in terms of what sequencing platforms are being used in the region, and this matters a lot when we come to supply chain, we found that, not so surprisingly, Illumina is the one that's most used. Second is ONT. Third is Thermo Fisher, and then Sanger is not necessarily a second generation NGS, but there's a lot of use of Sanger in the region for certain applications, so we put it in there. And then NGI, BGI is also becoming more and more popular. Now, this is for pathogens. If we redid this for human genomics, it would look somewhat different. So we haven't put in here PacBio. We haven't. Thermo actually has a much larger sort of market area for human genomics. So this would change a little bit, definitely, for human genomics. But these are the big players anyway, including PacBio, which in Asia for pathogen genomics at the time was not a big brand. We then broke it down by which countries at the time were using which technologies, and you can see Indonesia, in fact, was one of the few countries using MGI BGI at the time. So Indonesia, we realized, was willing to try out different technologies, which was interesting because we were keen to learn about MGI BGI at the time, and Indonesia had it. Different countries have different types of technologies. Now, this is great to see because that means there's competition in the market. But at the same time, if you're a bioinformatician or a laboratory person, you will realize that the laboratory workflow and the bioinformatics processing for each of these is different. So it does cause a headache, right? When data from all these countries are coming from all these different sequencing platforms. But obviously this is what is happening, and this is fine. This actually leads to competition in the market. Now, when we talk about setting up advanced technologies in low resource settings, one of the major issue that actually going in as a scientist, I didn't realize this going in, because I mean I'm always thinking about the science, right? But going in, we realized one of the major issues is supply chain. Even if the know-how is on the ground, even if they have the funds to do the sequencing, even if they have the equipment and the samples in the lab, they don't have the reagents because it is not coming in on time. They don't have people that can engineers that can come in and fix or do maintenance or fix their equipment. So we realized that this wasn't just in one country, it was happening across all the 13 countries, Asian countries we were working with. And essentially you can see it's also not specific to one technology, it's happening across the different technologies. And that has to do with the reagent and consumables purchasing lead time, having reagents and consumables in stock, equipment purchasing lead time, even custom clearance becomes an issue. A lot of the times they were complaining by time reagents clear customs, essentially the cold chain is broken. The reagents are no longer frozen, right? So the reagents can no longer be used. Then the the reagents, especially for sequencing, um, for sequencing reagents for some reason seem to have very short expiry dates. And when the supply chain takes very long, when it takes a very long time to get to the country or the lab, essentially they get reagents with essentially 2 months before expiry. And in research labs, I know we tend to use the reagents anyway, but this does cause a big problems if you're in a national public health laboratory, if you're in a clinical setting, etc. So essentially, expiry date of reagents on arrival is a big one. Distributor responsiveness. So we find that in a lot of high-income countries, we tend to have offices of those sequencing providers in country. So for Singapore, we have an office from Thermo. We have offices, ONT offices. We have BGI, MGI offices. So a lot of the times if we have a complaint, we go to them directly and we say, listen, we have this issue. But we find that obviously in the rest of Asia, it's really distributors that are doing the majority of these services. And they're not all, depending on, there are very good distributors, but again, there's a variety in how the quality of distributors as well. And obviously cold chain is a major problem. Oh, hold on, my slides disappeared. Give me a second. Okay. We then ask how long does it take for reagents to be restocked, right? It depends on the country, and on average, it took about 7.5 weeks for countries, for certain partners or labs to get their reagents. Now, maybe is this a normal timing for you guys? So whoever's in the lab, just raise your hand if this is a normal timing. 2 months roughly for reagents to come in from ordering. Okay, there's one hand here. Okay, so this seems normal, right? Now, okay, so I was trained in the US. Now, in the US, for example, I'm just telling you the difference. When I was in lab in the US, when I had, let's say, a PCR, I'm doing a PCR in the morning, I'm planning a PCR, I'm like, I'll go in reagent. I'll be like, oh, I'm missing a reagent. Either I will call the company and get it the next day, or I will walk down. I was in these big university campuses, walk to the next building, which has a big freezer university store, and pick out what I needed, and come back and continue my experiment. So that's the difference, right? Because the supply chain is there in those countries. In Singapore, it's a little bit longer, but we can still get reagents like in 2 weeks, 3 weeks time. But this is not the case for many countries. So these are things that we have to keep in mind when we're setting up advanced technologies in low resource countries settings, turnaround time. Now, financing is a big issue. We found that when we looked in how how was SARS-CoV-2 financed at the time? In many of these countries, 57%, almost 60% was externally funded. About 32% was public funding, right? So that means that if the external funding goes away, if the priority of the external funders change, that 60% goes away from our region. No longer is there funding to keep sequencing, right? So that is a problem. We found that financial sustainability is a big thing. So 23% of countries, okay, so if we flip the numbers here, because my director at the time didn't want the numbers to sound negative, so he wanted to sound positive. 23% of the countries have low reliance on external financial support. That means 77% has reliance on external support, financial support. 17%, only 17% considered that they had sufficient funding, and only 8% of the partners said, or countries said, they had sustainable funding for sequencing. So this is a big problem. So even if you have the capacity and the certain, let's say, equipment, you have technical ability, if you may not have the financing to keep this going. So when we set up sequencing or genomics capacity in low resource country or settings, we need to be mindful that we need to have some sort of financial source to keep it sustainable. Unfortunately, this is reality. I mean, this is something that came up in 2021 specifically for pathogen genomics, but this hasn't changed in a big way even now. Whether it's human genomics or pathogen genomics, the reality is that if you look at here on your left hand side, essentially, or right hand side, sorry, US, so high income countries tend to pay the least per sample for their sequencing. And income countries tend to pay double, triple, 10 times, 20 times more per sample for their sequencing. So these are countries that are already tight on budget, but they're paying way more. And some of that we've had discussions with the providers, vendors, and it does tend to be, some of it tends to be because, you know, vendors only mark down pricing if you're buying bulk. And if you're in certain, you know, if you're in lower resource countries, you're not going to buy bulk, these reagents in bulk, right? You only buy what you can finish in time. So this has become a bit of an issue. We did find out when we looked at other cost drivers, so what else is costing? Obviously supplies and consumables, equipment, maintenance. We do find that staff training costs do come up as a big one. So computing infrastructure is costly as well. Transportation of samples. These are the areas where a lot of the cost is going. We then asked questions around national planning and guidelines. The reason we did this is because if an advanced technology is to come into a country and truly be supported in terms of infrastructure, there needs to be a strategic plan, right? Obviously for national surveillance there needs to be a strategic plan, but even if it's human genomics, if it is supported by your health ministry and so on, if there is a national strategic plan, then there's at least something that can be implemented at the ground level. So we were sort of checking, are there plans in some of our countries, are there technical expert groups? Luckily about 69% had technical expert groups, and this is great, but this is also right out of COVID. So whether this still remains is a question. Many of them had guidelines for pathogen genomics, about half of them, but only about a quarter of the countries actually had a strategic plan on what to do for pathogen genomics and how to actually utilize it. Okay. Then we asked about major barriers, and again, finance is the key one for major barriers, and human resources. So lack of sustainable financing and lack of trained human resources. So not so surprisingly, when we asked about priorities, it was the inverse. They needed a lot of training, and they needed a lot of laboratory computing infrastructure. This is just a summary score that we did just pathogen genomic surveillance, and you can see sort of the capacity taking into consideration not just equipment and technical capabilities, but also national guidelines, supply chain, etc. And you can see the score changes with different countries. Actually, this work is published, so you can find it online, and it's on the slides. So I'm sort of gonna, this is my last slide, and I want to go through the summary. And essentially, this is a use case for pathogen genomics, right? How to set it up in low resource settings. However, this tends to hold true for other advanced technologies when you take it to a low resource setting. One, financing. So we need to make sure there's sustainable financing. Second, having a plan, where is what policy and guidelines come into play. Supply chain, making sure that the vendors, sort of getting vendors, discussing them, interacting with them, getting them to ensure that there is a way to get a stream of supply into your region. Some countries are looking into procurement purchasing, and that has worked, also to bring down pricing. Some countries or regions are also looking at regional supply chain solutions. Laboratory capacity, and I think that's Asia PGI tends to do a lot of this in terms of training, but also, this course happening here is also very important because we need a lot of capacity building. And here we've highlighted the laboratory capacity building and the bioinformatics, but we are slowly beginning to find out there's a need for capacity building and training in terms of multi-sectoral, especially for One Health issues. Also capacity and training where we bring individuals from different spheres together, as we do in this USU course. And I think that is my last slide, and that's it. Thank you, everyone.
Okay, is there any question for Dr. Ruki, please? Okay,
I think everyone's ready to go.
to go to Samos here. Okay, can I ask a question about the external quality control that you mentioned? How do you suggest the low-cost external quality control? That you know that sometimes the external quality controls should be standardized, and you can think that you have to send it to many far lab. What is your suggestion to make it low-cost acceptable?
So I think that's a good question. I think regionally in our Asian region, this is something that pops up over and over again. And actually, just to reiterate what Dr. Raza said, a lot of the time, mainstream what's happening is for external quality assurance control, we tend to send it to, let's say, a high-income country, right? And that costs a lot. But there is some discussion around sending something in Asian region, maybe a regional solution. It hasn't happened here, unfortunately. This is the problem. It has sometimes happened per pathogen. So I think WHO used to at some point, some pathogens like listed with used to provide certain
it's high cost. Yes.
Is there other question for Dr. B? Is that a question?
My name is Muhammad Jum'at and I'm in biology background. Actually, not too familiar in genomic sequencing, but I want to ask about how can genomic data be integrated into existing public health system in the research region without overwhelming the infrastructure or the capacity? Thank you.
Integrating the genomic results into health system.
Health system. Okay. Okay. Good question, especially like the one that Dr. Devi showed, right? The Satu.
Yeah.
Currently, interestingly enough, there's no. I haven't heard of a national health system that integrates their genomic data well into whatever national health system they have. You're absolutely right in the fact that pathogen genomics makes it a little bit easier, but for sure is too heavy in data. It just takes up so much computing hard drive space that it's a little difficult to make that. So that's a question that is or effort that's ongoing. How do you make sure that your genomics data, whether it's human genomics, pathogen genomics, gets integrated into your national health system? Currently, I don't know of a country that has resolved this in a big way. I don't know if Inca knows, I don't know if you know for human genomics, but pathogen genomics there hasn't been a solution yet. Currently, the sequence is lying in a separate database, so it's not integrated into, even in Singapore, it's all in separate databases. Yeah. But someday.
Thank you very much.
Thank you.
Okay, I think there's no more question. Can we give a big applause to
Satu, dua, satu, dua. Oke.
I guess with that, I think we can close the last session in the back. So everyone, I will invite you to have your lunch now, and then in 30 minutes, if you need to go for a Friday prayer, you can go for the prayer, and then we will meet again downstairs at 1:15 for those who are going to the semester. Thank you everyone. Assalamualaikum.